All right. Well, we have a terrific panel. You've already heard from Hannah about Fiona, Alison, and Pushmeet. We're representing here academics, life science industry, and IT world. And maybe just a quick intro. I think digital biology, or the large language life models, LLM, is moving at a pace we've never seen. Just last week, EVO was published in Science. This Wednesday, the Human Cell Atlas Foundation model will be published in Nature. We've had multiple human methylome models published in the last couple of weeks. Boltz one yesterday. It's just dizzying. I've never seen anything like it in my life. And so we're going to get into this. That's, of course, the kind of life science side. And, of course, is much broader, as already introduced by James and Jennifer, in terms of the biomedical application. So maybe we can just start off with where are things now. And so, Alison, maybe you can lead us off.
Okay.
Your experience.
Okay, so I work in imaging, particularly sort of ultrasound imaging. So I've particularly seen over the last 20 years how things have dramatically changed. Ultrasound is a great example where humans find it very difficult to acquire data, interpret data, and it's all about understanding the physics and acoustic patterns. So along came AI, and it's really transformed what you can do with an ultrasound device, because you can now, provided you've got the very large data sets that are required to do this, you can now find things in ultrasound scans that humans find difficult. And you can also guide people in how to use an ultrasound probe. And this is still emerging as an area, but I think is probably one of the most exciting breakthroughs in ultrasound, because you no longer require to be able to train for a long, long time. You can now put that into the front end of the device to be able to guide you. So that's just as an example.
Sure. Fiona?
Yes, thanks. So obviously heading up research at a pharmaceutical company, we're really using AI now right across the R&D process to advance new medicines, starting off with identifying new targets, which comes down to understanding diseases better. And that's where you use big data sets like the Cell Atlas, but also things like human genetics data, population data. The second area is once you have ideas for targets, how do you develop a medicine? So that would either be a small molecule, so that's chemistry, generative chemistry, and we use technologies like AlphaFold to design molecules, optimizing biologics or xRNA gene editing strategies. A very important area is in predictive toxicology. So making drugs safer as they advance, and then in clinical trial design as well. And there's a lot of innovations there using AI.
Sure. Pushmeet.
So, I lead the science team, and it's very difficult to sort of talk about what is happening because it's just so much. Across the board, I mean, if you look at topics, I can start with biology. I mean, James already mentioned the AlphaFold and AlphaMessen's work. I mean, really, what we are trying to do is decipher the genome, really understand what's encoded in the genome. And as Jennifer was mentioning, like if we have that amazing understanding, it will just accelerate the amount of work that we can do by using that amazing understanding about what is the actual semantics of the DNA. Now, if you look across the fields beyond biology, in every field we are seeing a transformation. James mentioned our work on weather prediction, GraphCast. Just to give you an idea, GraphCast makes predictions about near-term forecasting. It used to take supercomputers, simulations for days to come up with a prediction like that. And now it can be done on a single chip in a minute. That's like multiple orders of magnitude sort of difference. The same thing in materials. There are around 30,000 inorganic crystals which we knew were stable. In our work on graph neural networks for material exploration, we showed that there were 2.2 million inorganic crystals which were considered stable that scientists did not know about. And we shared 400,000 of them last year. And scientists now have discovered that many of the candidate materials that we had listed are candidate materials for high temperature superconductors, for better battery design materials, and so on. So in every area of science that we are looking at, whether it's biology, structural biology, genomics, protein design, weather, it's just transforming what is out there.
No question. I mean, Pushmeet and I were talking before we get started this morning about how structural biology used to be regarded as kind of the physics of life science, and now it's, because of AlphaFold and many other models, it's just, and of course, its use during the pandemic has been transformed. So this gets us to this acceleration of discovery, which is, I think, a theme of this conference, and how the science can be changed in almost a way that was inconceivable. In many ways, this brings up the role of the scientists in AI. And now we've seen things that started with the lab in a loop concept. It's been fostered by Aviv Regev, and then the AI scientist or co-scientist. Most recently, just last week, James Zoe published a preprint from Stanford about the virtual lab, where he had 5 domain agents that actually conceived nanobodies for SARS-CoV-2 that were very potent independently, essentially, from a infrequent human supervision. And that's, of course, going from virtual cell to virtual lab. So the limits here are something we've never seen before, some level of autonomy. So maybe we can start off by, this is something that we've not ever actually experienced, and it challenges our thinking about the future of science and the hypothesis generation, creativity potentially that we can derive. Who would like to start us on that path?
Well, I think it comes back to, we're moving so fast, we've got to be careful. It's great to hear about all the excitement, but the notion of what a scientist is changing. And we need to also stand back and reflect on that. In our report that we published, which was discussing with many scientists, in many, both AI scientists and across different areas of sciences, this is people's concerns. And it goes back to things like reproducibility, just basics, fundamentals. You had the slide at the beginning of the meeting where people used to get round a table and wanting to see the recipe repeated. Some of the principles haven't changed. And we need to, as scientific communities, demand to ourselves and in different research communities that we really understand when we publish results, that they are sound. Mm-hmm. That is partly thinking about openness. Openness I use deliberately because you can't make everything open. If you work in healthcare, your data is often sensitive. But that doesn't stop people maybe thinking of how they publish in different ways. Some communities are doing this, where you now are required to publish. If you can't publish your data, you have to be very rigorous in how you've evaluated what you do. So this is a responsibility for scientists to rethink in the age of AI, how we do present our work, and also how we do training of scientists. And not everyone is an AI scientist and maybe understands the methodology. So how do we train basic scientists to be able to trust the outputs? And another thing is the outputs. An AI makes errors in different ways to a human. Mm-hmm. So we've also got to understand the implications of that as well. Yeah. So we've got some wonderful early examples, but I think these are the sorts of issues we as a scientific community and in these areas we need to really understand a lot better.
Fiona, maybe you could also take us through a bit on the teams at Novartis. And
yes.
Are they into this, or are they have trepidation?
So I think this is a very important point to bring people with us. That's one of our priorities. So we have more of the concept of human in the loop rather than the lab in the loop. And how can we really enable everyone to embrace new approaches? And there's a number of ways of doing that. I think making everyone's job much easier by using AI tools, making them accessible so that people immediately see it as a benefit rather than a threat. That's one of the things. But also I've changed some of the hierarchy of how we organize our labs. Historically, the wet lab scientists generated data that then the data scientists had to sort of analyze and feed back. And they would be sort of center of the authorship on a paper, shall we say. But now I've actually put the data scientists as the lab heads in some cases, and so they're really the super users of computational means, and then they're sort of dictating the experimental procedures and the experiments to the wet lab scientists who then carry out those experiments. So that changes the way people think about data and use it, and is the fastest way of really sort of driving change in the organization.
Pushmeet.
So I think there are two sort of elements to it. One is AI systems as tools, and they are very powerful tools. They can make some things, they can make predictions which are quite unique. And as was mentioned, Alison, it's really important to understand when they are making progress and when they are correct and when they are wrong. And I think the real breakthrough of AlphaFold was not just that it was very accurate in predicting the structure of proteins. It was also that it was very accurate in saying, I did not make the right prediction when it did not make the right prediction. So the uncertainty, the predictive uncertainty of AlphaFold was extremely well calibrated. And that gives confidence to people who are using these sophisticated tools that they know when the tool can be trusted and when the tool cannot be trusted. Now, this is not the case for all the machine learning models out there. In the case of LLMs hallucinate, and one of the grand challenges is that you don't know when they have hallucinated and when they have come up with something remarkably intuitive and profound. So one element of this is, as you're using these powerful tools, how do you build the scientific community and how scientists can understand how to leverage these tools to the most effective manner? Then there comes to a point where you're using the AI systems in a more autonomous fashion. And this is what we are seeing when we are in our fusion projects, in our astronomy projects, where you have these controllers now which are running fusion reactors. And they are stabilizing plasma configurations in a way that has not been designed by hand. It has been learned. And so people who are working in those systems, they are extremely sort of cognizant of the safety implications. They want to understand what is the out of distribution generalization behavior of these models. So validation again is extremely crucial. When you give autonomy to such a system, you need to understand what are the limitations, how do you sandbox it, how do you properly test it, how do you exhibit, how do you test all the different ways the systems can go wrong. Right. So I think both from a tooling perspective as well as from a autonomy perspective, there are interesting challenges that we are actively sort of seeing the whole community and us focusing on.
Sure. I mean, the idea that was emphasized by Dame Julie and you as well, Alison, about this trust issue. Oftentimes, even the experience with chatbots is you tend to accept they assert, and you think this maybe is truth in fact when it certainly isn't. And also, I just think one case in point in genomics, a space that I work in quite a bit, because as James mentioned, one of the greatest challenges in human genomics today is that variants of unknown significance, VUS, or like UFOs, we don't know what they are. And in fact, the jump from from alpha missense to get to such a high number of variants that we understand more, not that it's absolutely right, but the fact that it's an order of magnitude increase in essentially decoding or understanding what variants of potential pathogenic significance. Now, one of the things we want to get into is this data shortage. So in talking with John Jumper last night, I think he agreed that Helen Berman and the Protein Data Bank, if it wasn't for them, there wouldn't have been AlphaFold. And we have limited resources of data sets. You mentioned, Pushmeet, the GraphCast, which the weather, we got lots of data on weather. So the problem we have now is limited great data sets. So we have this path of discovery, but we're missing the inputs. So maybe you could help us. How are we going to get these inputs, right?
So I think that, this point is extremely valid, right? If we are thinking about developing intelligence, intelligence does not arise from a vacuum, right? It arises from some raw material, and that raw material is experience. Now, that experience could be in the form of data that has been collected by scientists in a lab, or that is experience that the model has achieved by interacting with a simulation.
Mm-hmm.
And so we see both these elements working with high fidelity simulations, which can give the model a lot of intuition about what is going to happen. Like in the case of fusion and many of our physics models, we do use a lot of simulated data. And then, of course, there is experimental data like the PDB and the work that we have done in alpha-missense, which is also extremely sort of crucial. There is the third element, which is synthetic data, which is where you are trying to augment these data sources by making predictions. And distillation as a concept, where you try to make predictions about unlabeled data and then use those labels to train a model. And in the hope that some of the right predictions will give you intuitions and the wrong predictions will be thrown out, right? But that's sometimes can be wishful thinking. And it's really important that when you do these things, you have a very pure and firm test set, which keeps you on the ground and keeps you grounded on the fact that you are actually making progress.
Yeah, so I'd like to mention two examples here, because I think you've hit upon a very important topic. So in the context of drug development, and the first one I mentioned earlier is the predictive safety. So if we generate a compound or a biologic, can we predict whether it's going to be safe before we take it into humans? And of course, all the pharmaceutical companies have years and years of historical data that they've done testing in rodents, for example. So what we've done, and this comes in by way of histology slides. So glass slides in huge cabinets of drawers. It's like the Amazon warehouse. So, very difficult data to search that. So essentially what you need is to digitize all of those glass slides and then load up that data. And by doing that, you can then develop predictive algorithms for certain types of toxicity. So certain organ tox, what would produce cardiotox or renal tox. Now, we've decided that that type of data is not something we should just keep internally. I mean, this is best shared across the pharma industry to lift us all up. And so we've partnered with a company called Deciphex they're doing all of the scanning, but they're going to develop the algorithms, but they could also partner with other pharma companies, and then everyone will benefit from it. And I think it's one of the wonderful things about AlphaFold is everyone having access. So the more we can share data, the better. The other area which I know you're excited about is population data, population health data. So in the UK, recently we have Our Future Health, which is an initiative to get NHS data. Again, one of the benefits of the UK is having the NHS with all of this actually consistently stored, and nomenclature is important. How do you actually describe diseases? What do you call them? So again, this is a public-private partnership with universities, the NHS, and pharma companies contributing. And I think that produces an incredible amount of data to mine using AI.
I'm so glad you mentioned it, because first of all, we rely on the UK Biobank with 15-year follow-up, 550,000 participants. No data set we have of people is compared to that. And now I was astonished at how quickly Our Future Health is. Over 2 million people, participants, of the 5 million in just a short time.
With their records. And
it just makes all of us in the U.S. a joke, because you're going so fast, and you have, unlike the NIH, they're unwilling to have public-private partnership.
Yes.
And so you're doing that, and it makes a big difference. So we're relying again on the UK to have data sets assembled. Before going back over to Alison, I just want you to comment, as you mentioned in our discussions, people think of drug discovery, AI, acceleration, just in the molecule side, but there's so many different ways. Yes. Could you comment, for example, on just in the design and enrollment of clinical trials and other nodes of
Yeah,
this?
so we're using AI to help us run clinical trials. This is actually the slowest part of R&D. So designing the molecules, we are getting pretty good at. But once you have the candidate molecule, that can still take 10 years to actually get a marketed drug. And it's all the slowness of running the clinical trials and generating data. And so one of the key things is, how do you enroll patients? And using huge databases that have all of the demographic data of people around the world. Let's take the US. So where do people live who have certain types of diseases? What is their age? What other drugs are they taking? What hospitals are they associated with? By having all of this data, you can write the protocol for a clinical trial, and you can enter different parameters. Like, I would like women age 30 to 50. How does that affect the enrollment rate? I don't want them to be taking this drug. So we talked about breast cancer, whether they would be taking, estrogen pathway modifiers. How would that affect the enrollment rate? And by optimizing then all of the parameters, you can massively accelerate the recruitment of a trial. So something that might take you 3 years to just recruit the patients to run the trial, you could now recruit those patients in, say, 6 months. And this is a huge acceleration of getting drugs through to the patients and to market.
Yeah, no, it's phenomenal. Alison?
Just partly listening to what's also said, we've talked a lot about partnerships. And when we talk about the culture change that's going on, this is dramatic in academia, where traditionally people would have their own, they'd work on their own in their small lab, they would be highly competitive with everybody else in the world. Along comes a tool, the family of tools of AI, and you now want to be able to have the data to be able to answer questions. You've suddenly got to partner with everyone. And this is proving to be quite difficult in certain areas. And this notion that competition drives innovation, you've got to balance that with how do you manage partnerships as well. And also in academia, how do you manage rewards now? The classic reward system in academic science would be to publish papers. And now you have many, 20, 50 people can be on a paper because you've been because they've all contributed to a particular idea. But how do you make it attractive for young scientists to enter a field, be noticed, and their careers to progress when you work in this interdisciplinary area that's team science? And I think this is something else we need to have. New ways to reward. It's not new to suggest it. We've had the idea interdisciplinary research is always a challenge, but I do think we need to think about the new ways that we can recognize people.
Absolutely. I also want to take the chance to zoom in a bit on the ultrasound work you're doing, because a lot of it was brought up in the inspirational video that started this morning about reducing inequities, and the idea that you could obtain images through things like a smartphone anywhere in the world, even by people who have no training in capturing images. Could you comment about that?
Yes, because I've some experience of doing that, of taking the very low-cost probes. They're sort of under $1,000, and the idea there is that you will be simplifying what a person does. So sweep across the pregnant mum's abdomen, for example, and then let the AI decide what's there, what might be the issue. And these sorts of, we've both developed some early technology to do this. We've also, as part of the research, gone out and asked in Kenya and also in India, would they find it acceptable to use tools of this kind? And the answer is they're like everyone else. They want to know that tools work, but otherwise they would very much like to use them. But it's a very different way to do healthcare. You've got a different, you mentioned human in the loop. The phrase I use is worker in the loop, because whoever the person is holding the probe, you've got to empower them. And that means that you might have different capabilities, different explanations come for what's found. Are you, for example, using it for triage to refer in, or is the decision being used for actual decision making? So it's really an early emerging area, but it's a fascinating area as well.
Yeah.
But the notion that everything needs to be safe, but understanding that worker is really key to see adoption as well.
Right. And one of the things that's really different about life science and the models that are cropping up now and changing things at a pace that we've not ever seen, whereas in medicine, there's this thing called regulatory. And this, of course, has been very difficult because life science can move much faster. It also has the ability to really take advantage of simulations and synthetic data in a much better way. So maybe you could comment. We talked a bit about there's a worry about using too much synthetic simulation data. And what's your perspective about how we can have comfort about that?
Yeah, so I think at the end of the day, the proof of the pudding is in the eating, right? So whatever the recipe is, whether you have synthetic data or whether you have data that has come from a simulation or from a wet lab. And by the way, I should say that not all wet lab data is equal. We have seen even data sets like PDB was amazing, right? And there was a particular sort of quality threshold. But if you look at data sets in other areas, the quality level is not the same. So it's really important to focus on having good test sets to really which tease out how much progress is being made. And in that sense, AlphaFold, one critical aspect was the PDB, but the other critical aspect was CASP. That CASP was a really good signal for us to track whether we are making progress on out of distribution generalization and really sort of and predicting things which are currently not known. So investing in test sets where there is no risk of overfitting, you are taking out a time-stamped view that the model has been completely shielded from the things that it will be evaluated on is an important element of the testing protocol that we use.
Well, you mentioned CASP, and one of the things that's very different in life science is that you have these annual contests.
Yes.
And we don't have that in medicine, right?
Yeah.
So does that really help the competition, that managed competition, bring out even better science and creativity?
Yeah, I think like the critical assessment, CASP is critical assessment and structure prediction. And for those of you who don't know, I think it was an amazing and very visionary idea where it was run sort of biannually, and the competition was that any sort of new structure that was experimentally found out for a specific period of time, scientists would deposit it in a vault,
Yeah.
right, and will not share it with the world. And these sort of structures were then kept hidden.
Yeah.
And the whole community of structure prediction, people who are working on structure prediction, would be told, okay, here are the things that you should test on. And now nobody knows the answer, but the true answer is known, right? So there's no way to cheat. And so that gives you a very clean signal of how much progress is being made, and you're not fooling yourself by just overfitting or trying to sort of think that you're making progress, but you're not.
Yeah. No, it's.
Eric, I thought you'd be interested to know how we're using AI for regulatory part
Yes, I am
process.
very interested in that.
Basically, every single regulatory question and answer is mapped on a database. And so now it allows you, as you submit the regulatory documents, to predict what questions are going to come up by the regulatory authority. So basically, you could be well prepared. But then even better is once you get the sets of questions that come in, you can find out, have those questions been asked before, and then predict what's the best answer to give that's likely to give you a positive approval for your drug.
That's terrific. I do want to get back to you on this because one of the greatest delays of getting drugs approved is having to submit what we call it
thousands of questions
IND.
Yes.
It can take a year with
Exactly.
hundreds of or thousands of pages.
Yeah.
Do you think INDs will be AI generated?
Well, that we're already using them.
You're
to
already
generate-
doing it.
yeah,
Okay, there you
absolutely.
go.
I mean,
There
of
you
course,
go.
they do have the human in the loop.
Yeah, but it could
they're
be-
massively becoming AI enabled through the whole writing process, and the whole process then of going
Yeah,
through
so I mean, the
regulatory-
acceleration isn't just the molecule. There's a lot of other features, as you
Yes.
mentioned. I didn't realize you already were onto that. Wow. Alison, do you want to?
Yeah, I
weigh in on
can.
this?
It's interesting that you get the audience going when we talk about regulatory. So there's regulatory approval, and then there's the purpose of regulation. And I think that there is, if I think in my area, if you extend it to things like radiology, it's very important to have the regulatory approval. But then, of course, that's not telling you how these systems are going to be used in practice when they're deployed, and also the human behavior changes that can come when you use an approved system. So this is an area where I think there is still some more work, because demonstrating it's safe is one thing, demonstrating that and understanding how human behavior changes in a clinic, because you've now got a tool that you might then become dependent on. You might trust it too much. Or you might switch it off because you don't agree with it. These are the sorts of things and the studies coming out now, which are a very different extension to is it safe to use in an environment. And I think the other thing is, as some of the AI tools become, and if they are better than humans, it's very different than if they're supporting a human in making decisions. And we need to understand more about the implications on that and how it changes decision making.
In that light, I want to ask you about there's a lot of debate about the ability to be creative and reason for AI systems. Where do you come out on that? Is this just a function of pre-training, or is there really is this dimension of thoughtfulness and critical reasoning capability?
In the domain I work, I don't think you would say it's creative, but maybe there are people working in spaces where that you're finding the gaps where the humans don't understand that particular space of the data, but it's all driven by the data. So it's very exciting, I think, when people can discover something and then they can attribute that to being creative. But I would say it's modelling gaps that the human brain hasn't thought about.
Okay. How about you?
No, I mean, I think this is why you need the humans, because it's that feeling of empathy, understanding. If you know somebody who's dying of cancer, that deep empathy that you have for that person and wanting to bring a new medicine for that person, I think I can't imagine a computer being able to have that level of understanding.
Okay, now you may come out differently on this, Pushmeet. I'm curious. What do you think about the advanced capabilities for reasoning and?
I think. See, there are, if you think about AI and if you think about reasoning at different levels, there is reasoning at a non-expert level. And there was a dramatic progress that AI made in things like perception. That's not something that an expert, we all can basically perceive the world and can do object detection, image classification, but it was a big breakthrough when AI systems were able to do it. They were able to achieve non-expert level reasoning behavior. Then there is expert level reasoning behavior, where you have a very sort of a person who have spent a lot of time in understanding a particular field and getting the real sort of facts out. And then there is beyond expert level behavior, which is what systems like AlphaFold and AlphaMissense do, where there are no humans that are able to solve that problem. So I think if there is these models are able to do these reasoning problems from the input that they have. If there are no signals or there are no input features that they can sense from, then they will have that fundamental limitation. So if they have the ability to perceive how someone is feeling, then they might be able to sort of reason on top of that. But if they are fundamentally, the interface is primitive and you are just talking to them by textual means or by saying, here's the sequence, then of course, will have fundamental limits on what you can do with that limited information.
Well, I think that's I think a good stopping point in that we're still exploring this topic and will be for some years to come. And we see hints of it. We've seen in medicine how promotion of empathy consistently across many studies, who would have ever guessed, because of just understanding language. But there's, as I think James emphasized, we're in the early days across all this. Just think where we'll be when this meeting convenes in 3 to 5 years. So let's thanks so much, Alison, Fiona, and Pushmeet, and thank you for your attention.